Today I'd like to present you an innovative solution to break the wall of incomplete recovery following nerve injury. And to understand the relevance of nerve function for our daily life, we need to realize that any movement and muscle contraction, any perception or sensation in the human body is transmitted by nerve fibers. These nerve fibers bundle up to cable-like nerves that traverse our body and connect our muscles to our brain. These cables are vulnerable. Even a small piece of broken glass can cut these nerves and interrupt the crosstalk between brain and muscle, resulting in complete loss of muscle function. Fortunately, injured nerve fibers have the amazing ability to regenerate and reconnect to the muscle. But while the nerve fibers slowly regrow, the still disconnected muscle itself starts to degenerate. This frames a race against the clock as the nerve fibers try to reach the muscle before functional reconnection becomes impossible. Unfortunately, in many cases, the nerve fibers lose this race, with devastating consequences for the patient and our economy. Following upper arm nerve injury, as for example, 1 in 4 patients loses their job, and every second patient suffers from lifelong disability. Despite these devastating consequences, the treatment strategy for nerve injuries remained largely unchanged for over 70 years now. This is because the drugs that are known to boost nerve regeneration have systemic side effects that prevent their clinical use. As a result, to this day, there is no treatment available to support nerve fiber regrowth. We want to break this historic wall of nerve regeneration using the concept of local microdosing. We developed a polymer-based local drug delivery system to enable neuroregenerative therapy directly at the nerve repair site. This drug delivery system is a fully biodegradable, ultra-thin membrane that is wrapped around the nerve during surgical nerve repair. As the body degrades the membrane, it slowly releases microdoses of a neuroregenerative drug directly to the nerve over a period of up to 30 days. Using this novel system, we managed to overcome systemic side effects and increase the number of successfully regenerating nerve fibers to 99%. I'm presenting this today because I believe the Falling Walls Network will help us to bring this project forward and prepare for the next phase, the phase of the clinical trials. Thank you.
Thank you, Simeon. Yes, a round of virtual applause first. Thank you so much. And now we open the floor for questions. Please, the jury, I would ask you to raise your hand so that I can see it. So, Geeter is first.
Hey, you had FDA Approved Drug on this slide. What kind of approval do you have?
So the drug that I'm talking about is FK546 or tacrolimus. It has got its FDA approval like 25 years ago, but not for nerve regeneration. It's approved as like an immune modulator for example, organ transplant or autoimmune diseases. So there's an extensive kind of experience with this drug already.
Can we have a second question? No? Good, please.
What kind of partners would you need to go through these clinical trials?
So I think a next step would be that we would need to connect to the pharmaceutical industry in order to meet the production criteria for clinical trials, as for example, for human studies. And also we would need persons that have experience with the approval process for clinical trials itself. So currently we're like in a preclinical setting, in vitro and in vivo studies.
Time's up, I think.
Thank you, Sophie.